Hi everybody. Today we're going to be talking about blood splatter and serology. Serology, now if you don't know, ology means a study of, so S-E-R, I wonder if you could think of anything having to do with blood that starts with S-E-R. I'll give you to the count of 10. See, if we're in live in class, you'd be raising your hand right now. But if you came up with the word serum, S-E-U-R-M, then you hit it, okay? So serology is the study of the serum, or basically blood, right? And blood in all of its parts. Inside of forensic science, that's really important because we can, as we've seen with lots of other things, we can learn an awful lot about a crime scene based on blood. The objective in this lesson is we're going to learn blood splatter analysis and serology procedures. It's not really telling you all of it because before we get there we're going to learn about DNA. So now, as usual, I just want to remind you that I skipped over lots of stuff and I don't want you skipping over it, so I'll only go to the good parts in this video. Everything else you are responsible for doing, but we're going to start here, the fundamentals of blood and genes. So we're going to jump into a discussion about immunity a little bit, and then genes and genetics. So we're going to start talking about something called antibodies. Now, antibodies are also called immunoglobulins. Okay, that's a big word, but it just basically means it's a structure inside of the blood. You know, the blood's just not one thing. It's a mixture. It's got plasma and water and all kinds of dissolved minerals in it. Plus, there's I think 25 different types of cells, not just red blood cells. There's white blood cells and T cells and helper cells and B cells, and a lot of things have to do with your immune system. Antibodies are found in your immune system, and they're a response to foreign agents that get into your blood and your body. They're proteins that your body then creates to help fight off these foreign substances. Okay, so antigens can be bacteria, viruses, fungi, or allergens. That's what the antigens are. These are the things that make you sick. The antibodies are designed to fight them. They have a very specific shape. Mostly they're Y-shaped, and then the Y and the points of the Y are designed to actually stick to the outside of the bad guys, the antigens, and they neutralize it or deactivate it. A long time ago, they used to have this, not a long time ago, like 10 years ago, they used to have this thing where they could control riots. They would spray you with like this foam, right? And it would harden around you and you couldn't move. Either that's real or I saw it in a sci-fi movie, but either way, these kind of remind me of that. What your body does is it kind of floods when you get sick, it floods your bloodstream with these antibodies, and they stick to the outside of the antigens. And the antigens aren't dead, but they can't do anything because they're kind of covered in all these antibodies. And once they are deactivated or neutralized, then your body has processes for grabbing them and destroying them. But your immune system is very complex, but this is one of the most important parts. Real quick, they're classified into 5 categories. We have IgC, which I think this was supposed to be a C. So if you see IgG, it's really IgC. These are antibodies that circulate through the body to defend against bacteria and viruses. IgA, they're present in body fluids such as tears, urine, and other secretions, and they're to prevent viruses from entering the body. IgM antibodies are the frontline defense when fighting against bacteria or viruses. The IgC antibodies take over if the IgM antibodies can't neutralize, so they work together. They're like tag team wrestling, I guess. IgE antibodies are present in the watery part of body fluids such as mucus and tears. IgE antibodies cause allergic reactions to produce histamine, which removes allergens from the body. Let's talk about that briefly real fast. IgE antibodies cause you to have allergic reactions and produce something called histamine. Histamine then starts to work on those things that are allergens. Okay, the problem is that our body in this particular system right here tends to overreact. Okay, and these histamines cause us to have things like nasal dripping, sneezing, watery eyes, and swelling, all of it with the intention of trying to keep this stuff out. Except our immune system doesn't know when to stop. And so a lot of times when you have these things in your body, you get these allergic reactions like nasal dripping. You can get sick from it. It causes pneumonia. The sneezing, the watery eyes, they're no fun. And then the swelling is a really bad part. If you're allergic to something like a bee bite, you get bit by a bee, you can swell up to the point to where, and that's your body's reaction. The swelling is your body's reaction trying to deal with this new thing that this venom that the bee injected into you. The problem is your body doesn't know when to stop, and it can actually swell you so much that it swells your esophagus shut and you suffocate. So it's not a perfect system, but histamines are good. All right, and you think, well, I've heard of histamine before. Oh yeah, I take an antihistamine. Well, again, when your body goes too far, you have to have an antihistamine to keep that from killing you. And an epinephrine that's in an EpiPen is an antihistamine, and if you get in a situation where you have an allergic reaction, if somebody carrying an EpiPen, you pop that into them, and it may keep their immune system from killing them. So it's pretty crazy. I have been trained on EpiPens. I've been trained how to deal with this. My brother is allergic to bees, and so, man, I'm telling you, it's a pretty bad thing. All right. And then finally, IgD, or antibodies, responsible for identifying antigens and producing the antibodies to fight the foreign substance. So IgD kind of is the quarterback. It starts the whole process and it tells everybody else what to do. All right, now why do we study all this? What's this have to do with forensic science? Well, you're going to ask that a lot today, but trust me, it's important. Before we go into why all that's important, well, that's not true. We're doing it right now, but we want to talk about your blood type, which is kind of what we're leading to. Learning someone's blood type is based around an antigen, which is on the outside. And you say, wait, I thought you said antigens were bad guys. Well, okay. Antigens are just spots on the outside of the bad guy, right? It's like a node where you can stick stuff, okay? So you actually have it around lots of cells, some good guys and bad guys. And you have cells, your blood cells, for example, have antigens on the outside. So the antigens are just spots, but they're very specific, okay? And I do mean they're like having like having an ID badge on the outside of your shirt, okay? And your body looks and goes, whoa, that's a bad guy, we got to kill it, because it sees the ID, or it sees your ID and it says, whoa, you're a good guy, we're going to keep you. Now we have two types of antigens that we're going to talk about on the outside of our blood. We have the A antigen and the B antigen, all right? And then there's basically four types of blood. You have those people that have the A antigen on the outside. You have those people with the B antigen on the outside of their blood cells, right? Some people have both A and B antigens, and some people have none, okay? Well, remember, these are like ID tags, and the body can react to all of these ID tags if they have to, okay? So, for example, if you are an AB type blood type, you don't have any of the antibodies that would then attack the A or the B because you have both, right? So we're going to get to that in a minute, but if you're an AB blood type, you don't have any of the antibodies. You just don't have them. You don't need them because there's the A antibody and the B antibody, and you don't use them because you're fine. They're only there to attack the opposite, right? For example, if you're an A blood type, you got the antibodies for B, because you got guys constantly prowling around looking for B, going, whoa, anybody a B? Because if you got a B, like a name tag, if you got a B name tag, we're going to kill you. You're not supposed to be here. It's kind of like a gang, I guess. I don't know. So if you have an A blood type, you've got B antibodies looking for the B blood type, because if it gets in you, you don't want it in there, okay? Same thing, if you got the B blood type, you got the A antibodies running around looking for A. I don't want you in here. We're going to kill you, right? If you have both A and B, you don't have any antibodies because you don't need to look for anything. Now, if you're like me and you have type O, then you've produced all of the antibodies that go with it. The A antibody and the B antibody. I have no antigens on the outside of my blood, and I have a lot of antibodies running around looking for A or B. Now, back in the 1900s, a guy named Carl Landsteiner, I don't know if I said that right, was noticing that sometimes blood transfusions worked on people and sometimes they didn't. You get a blood transfusion, you're great, or you get a blood transfusion and it kills you. And he's tried to figure out why. And he started doing some studying. And he discovered all this. And he found out that there are some compatibility issues. And so we're going to be talking about these compatibility issues very briefly, but also understand that we can type blood now based on whether it's an A, a B, an AB, or an O. Now you may be thinking, well, wait a minute, I'm positive and a negative. That's an entirely different set of antigens. There's a whole nother thing on the outside of these that gives you positive or negative. So that actually gives us 8 blood types, but I'm not going to go into that. So take, for example, me. I'm type O. Let's just talk about donating blood before we get back to the forensics of it. I'm type O. If you give me type O blood, and my antigens or my antibodies are going to be looking around going, hey, anybody a bad guy? Well, are there any antigens on the outside of the type O blood? No. So my antibodies go, great. That's bad news for me because I can only take type O blood. If you give me type A, type B, or type AB blood, my body's gonna reject it and it'll probably kill me. Okay. Let's do let's do the opposite. Let's say you have type A blood. Okay. Now remember, you've got type A blood and antibodies looking for B. If I give you type A blood, are you gonna be okay? Yeah, because the antibodies are looking for B, not A. So now I've got new blood in there with the A name tag on it. Everybody's good. If you're type B and I give you type B blood, you're good. Now there's new guys here, but hey, you got the right name tag, you're good. All right, man, good to see you. But suppose I give type A person some type B blood. Now all of a sudden this antibody sees the type B blood and attacks it. And it creates a real problem. These are the weird guys. If you're AB, you're weird because you got both. You're what we call the universal receptor. I can give you type AB blood, I can give you type A blood, I can give you type B blood, I can even give you type O blood. By the way, do you see an antibody for type O blood? No? That's because there's no antigens on the outside, so there won't be any antibodies. This person, AB, can take any of the blood types, AB, A, B, or O. Okay? However, let's suppose we wanted them to donate blood. If they donate blood, they can only give it to people that are AB. Because if you put AB blood into someone with A, you may be thinking, well, they're okay because I got type A blood. But no, remember that antibodies run around and it will see that type B antibody on the outside of that blood cell and it'll attack it and try to kill it. So as a result, type AB is great if you have it. You can get anybody's blood and don't worry about it. But if you are trying to donate it, put this blood into anybody else, you'll kill them unless they're type AB. The opposite of that is me. I'm the universal donor. I can put my blood into anybody because there's no antigens. Nobody is going to reject my blood because if you're AB, you don't have any antibodies. If you're type A, you've got B antibodies. Do you see any B antigens here? Then no, this guy won't attack it. If you're type B, then you have A antibodies. Do you see any A antigens here? Then they won't attack it. The bad news for me is I can't take blood from anybody because I have the A and the B antibodies. And so if you give me any of these other types of blood, they're going to attack it and kill it. Now, this guy Landsteiner discovered all this back in the 1900s, and since then we've learned to blood type people. Okay, we refer to it as the ABO blood typings because there's A and B and O. And as a result, we can now tell you what blood type somebody is. And if there's blood left at a scene, we can type that blood. And like I said, I know there's 4 here, but if you do positive negative, that means 8. There's 8 blood types. And if we can figure out which blood type was found at the scene, it can dramatically reduce the number of suspects that are in at the crime. You know, well, he couldn't have done it because he's type A and the blood type we found was type B. Ah, says the jury. Now, you inherit your blood types from your parents. They're actually found on genes, which are encoded onto chromosomes. These are chromosomes. You know, you get chromosomes from your mom and chromosomes from your dad. You get 23 from your mom, 23 from your dad, okay? And they kind of match up. So we number them 1 through 23. And I don't know, does it tell you which chromosome it's on? It does, I think, but let's say it's on chromosome 19. We're just making this up. If this is chromosome 19 from your mama, this is chromosome 19 from your dad, okay? And on those, in various places, we call these alleles. In various places are the alleles for genes, for traits. The eye color trait is here. You got 1 from mom and 1 from dad. The blood type is here, 1 from mom, 1 from dad. The hair color is way over here, 1 from mom, 1 from dad. How much you grow is right here, 1 from mom, 1 from dad. Now you guys remember the whole dominant and recessive things? Well, this is how it works out, right? If I get a dominant trait from mom and a recessive trait from dad, guess which eye color I'm going to have? I'm going to have mom's eye color. If I get the reverse, I'm going to have dad's eye color, right? And it could possibly be that mom and dad have one of each, dominant and recessive, and they give me the both recessive. They don't have brown or blue eyes. My parents don't. But they both passed to me a recessive trait they didn't even know they had. Now I've all of a sudden got blue eyes. How do two brown-eyed babies, mamas, have a blue-eyed baby? That's because brown is dominant to blue. And that's exactly what happened with me. My mom and dad both have brown eyes, but I have blue eyes. And that's because they have the dominant and the recessive trait, and they both gave me the recessive trait. Very low odds of that happening, by the way, something like 1 in 16. So here I am with this. I think I'm wrong about that. I think it's 1 in 4. But that's all right. If anybody, biology teachers, are watching this, please forgive me. So how do we, why is this all important to immunology, or sorry, forensics? Because now we don't even have to have your blood type. We can just have your parents' blood type, and we can predict which blood type you are just by doing a Punnett square. Okay. Here's a Punnett square for me. All right. We're just going to pretend. This would be my mom. She has brown eyes. Okay. This would be my dad. He has blue eyes. Let's just pretend. Okay. What are the odds that the offspring is going to have blue eyes? Well, we can predict that with a Punnett square. Each one of these squares represents a possible type of offspring. All right. Now, mom had brown eyes, but what mom didn't know, or maybe she did know, was that she carried the trait for blue eyes. Well, now wait a minute. How come her eyes aren't kind of a bluish brown? Because that's not how DNA works. Mom's got brown eyes because she's got the dominant trait, but she's got blue eyes too. It's just not showing up. She probably got it from her mom or her dad. She got it from one of the two. They may have not had it either, right? But this woman and this man can produce two types of offspring. This square represents the odds that you're going to get a brown-eyed trait from mom and a blue-eyed trait for dad. That's what this square represents. This square represents the same thing. The odds that you're going to get the brown-eyed trait from mom and the blue-eyed trait from dad. This square represents the odds you're going to get the blue-eyed trait from mom, the hidden recessive trait, and the blue-eyed trait from dad. Boom, there you go. And then same thing here. Boom, there you go. Well, now wait a minute. How come this baby has brown eyes, not blue eyes? Because brown is dominant over blue. Now I hope you guys remember all this, okay? But it's pretty simple. With these two parents, there's a 50% chance the child's going to have blue eyes and a 50% chance they're going to have brown. That's how Punnett squares work. We can do the same thing with blood typing. If mom is A and A, then she's an A blood type. If she's B and B, then she's a B blood type. If she's O and O, she's an O blood type. She could also be AO. That's dominant and recessive. If she has dominant A and recessive O, this was my mom. My mom was actually dominant B and recessive O. What blood type does she have? She's got B because the O is recessive. My dad was AO. What kind of blood does he have? He's got A. But I'm that little guy right there. I got the O from my mom and the O from my dad, and I've got O blood type. Genetics are weird, and I know this was really fast. I hope that this is just a review for you. But knowing how Punnett squares work, and then knowing what we can do with parents and offspring and prediction, we can do an awful lot with blood typing and stuff like that to learn things about a crime scene. And here we go. For example, this gives you all of them. If the mom passes along an A and the dad passes along an A, you're going to get AA. So the offspring is blood type A. This is not a Punnett square, by the way, just so you know. This is just showing you all the possibilities. If a mom passes along a B and the dad passes along an A, guess what kind of blood type you got? AB. If the mom passes an O and the dad passes an A, you get AO. Now look, it's not like the mom or the dad has all of these, and it's just that any of this could happen. Any parent has two of these. They may have an A and a B, an A and an O. They may have two A's. They may have two B's. They may have two O's. All right? So you take the two parents, whatever the mom has, AA, AB, AO, whatever they've got, whatever the dad has, AA, AB, AO, whatever they got, and you put them into the Punnett square. So like I said, let's say that dad was AO and mom was BO. This offspring here, you got an A here and a B, so this would be AB. They got AB blood. This would be AO. They've got AO blood, so they're type A. This would be OB or BO. This would be B blood type, and then this would be OO, so they would be blood type O. So there's a Punnett square problem. That's part of the lesson activity that you have to complete. I'm going to help you out a little bit. I want you to notice some things. The dad has brown eyes, and the mom has green eyes. Okay, I don't know how to highlight all that, but I want you to think about something. Is that their phenotype or their genotype? Remember which one is which. So you got to dig further. You can't do a Punnett square knowing someone's phenotype. So we got to dig a little bit deeper. So it starts to tell you the genotype. The father has a brown allele and a green allele. Does that mean he's got one brown eye and one green eye? No. Brown is dominant over green. So the dad has brown eyes, but he's carrying a green eye trait, recessive. The mom has green eyes. Now she doesn't have two green eyes. She's got two green eye alleles. Okay? So she's going to have green eyes even though she's got two of the same alleles. Right? She has recessive. It's like having O blood. You got both recessive traits. Well, now the mom has both the recessive traits. She has green eyes. If dad has brown eyes but a hidden green eye trait, and the mom has green eyes, can we predict the outcome of the possible offspring? Okay? And then go ahead and down here and do a self-evaluation. And now we're going to talk about blood splatter. Doesn't this just sound fun? I hope you're not eating. So blood splatter in crime scenes are unique, and forensic scientists and forensic techs can learn an awful lot. Plus they can grab all this information, some samples. They can take pictures of the splatter patterns. They can grab samples of the blood and other stuff. We're going to talk about semen also, and they can ship it off to a lab and learn a lot. You can do a lot of stuff right there, and you can ship it off to a lab and do a lot of stuff. The video that I have included with this week talks about that quite a bit as well. The first thing we're going to talk about are three types of blood splatter. The first one is called origin, okay? But you can tell an awful lot about where the blood splatter came from by how it hits and how it streaks. And there's some math you can use and some other stuff, but this person or this particular blood pattern right here came from right here at this spot, and they were able to tell by looking at the little, how the blood hit and the little bitty lines like that guy right there and that guy right there. They can tell an awful lot about which direction the blood came from at a crime scene. Very important. Impact, okay, tells you whether it fell from the top down, such as dripping blood, or whether it was just kind of slung by some type of force. Maybe a person got hit and blood came out of their body. And then finally direction. Maybe not so much origin, but you can tell a lot about which direction something came from based on the streaks of the patterns. So when we look at blood splatter, there are three different types of patterns that we look at. The video I'm showing you with this unit is good about that. Read this and try to understand a little bit more about it. These three panels, they're very, very informative. And honestly, this slide is just a repeat of the last slide, almost word for word. But read these. You know, there's several ways to determine the origin or source of the blood. I actually think So you can use graphing, tangent function, triangulation. Of course, nowadays they got computers to do it. You just take a picture and the computer can do it, but you can still do it by hand, right? What is impact and direction? We've already covered all that. Now, latent blood stains are not visible to the naked eye. They're latent, which means that they are hidden, and you have to find them, right? Got a lot of ways we can do that. They're very important to find inside of a crime scene. You can use high intensity ultraviolet lights to find blood. It kind of reflects bright. You can shine it into a room and find that. You can also find other body fluids such as semen with the same kind of lights. Most investigators carry one around in their pocket these days. And you're able to find a lot more about it just by shining around a UV light. We've got one here that my wife uses for nursing, and we sometimes play with it, like if somebody gets cut, we shine it on the blood and see how it shines. Yeah, we're kind of weird, but There's 5 total. Another one is called luminol. Luminol is a chemical that once you spray it, it causes the blood to change in color a little bit, and you can see it better. It'll glow blue, right? It has its limitations. It reacts with substances that contain iron, and there are other substances that have iron as well, so it doesn't just show up with blood. Luminol can show other things as well. Plus, luminol destroys other evidence of the crime scene, so you have to be very careful with how you use luminol. And did I say there were 5? I meant 4. Fluorescein is another chemical. It reacts with blood. You spray it onto the crime scene, and then you wear ultraviolet light or goggles. It reveals a greenish white color. It has its limitations. It can react to some metals in some cleaning products. You can also damage the crime scene as well. And LCV, or leucocrystal violet, it's a chemical that makes blood more visible. Now, it does not light up to reveal blood. It simply makes the stains darker, so you can see it. You don't have to have a light or some other thing. You can just see it. It shines right then. And then you take a picture of it and send it to the lab. They all have their limitations, which is why we use all 4. And a good crime scene investigator knows which one to use at the right spot and how to not contaminate other parts of the crime scene. It's a bit of an art, and I hope you're not eating pizza right now. So the lesson activity that goes with this is the second activity. Make sure you're doing them all. Is to analyze some blood splatter. It's easy. Just take what you've already learned about blood splatter, go back and look at your notes, and just tell me something about figure 1, 2, and 3 that you might be able to tell in terms of direction, origin, and impact. I'm very curious to see what your answers are. Make sure they're pretty cool. And don't forget to do the self-evaluation. So the last part is analysis of body fluids. We're going to be talking about all kinds of analysis. There are 2 types. There's what we call the presumptive test. That's done right at the crime scene. It establishes, yes, there is a body fluid. We don't know necessarily what it is, so they do a couple of tests right there on the site. Yes, that's a body fluid. Yes, we need to grab some of that and send it to the lab, where they do what's called a confirmatory test. The confirmatory test is a much more specific type of test that can tell you a lot more about what type of body fluid is, even like giving you a blood type, for example. You can just look at the scene and go, yeah, we think that's blood. Put a little presumptive test in there and go, yep, that's just a little stick or something, maybe like a blood test strip, and you go, oh yep, that's blood, so we need to grab some of that and send it off. But at the lab, they can tell you, yes, what kind of blood it is, the blood type, etc. They'll sometimes even tell you, you might not know that if it's human or a animal type of blood. When we get to the lab, when we get the samples to the lab, they can run a couple of tests. The first one's called the Takayama. All right, it's less reliable with older stains, but it gives a very good results. The Teichmann test uses entirely different stuff, but it's reliable in different ways. Again, we have all these different types of tests because different types of tests are more useful, or they're more certain depending on the situation. Okay, so we have lots of tests, and we if we can afford it, we run as many as we can, because remember, we're trying to hand evidence to a district attorney who is trying to make a case, so we want to do as well as we can. All right, both of these test results can produce a result with 0.001 milliliters of blood, and I'm telling you, you couldn't even see that much blood. It'd be hard to see it with a microscope. That's how accurate they are. Okay. For a dried blood stain, you can get away with 0.1 milligrams, which is unbelievable. It's a dot. It's a period at the end of a sentence. That's how much blood it is. And yet we are that hyper accurate with these tests, which is incredible if you think about it, because just 20, 30, 40 years ago, we couldn't have done this. Another thing we can analyze is saliva. Saliva is often found at the crime scene. Even if it's dried saliva, it's still useful. So we can collect samples of saliva and put those samples into a test. Again, we can use presumptive and confirmative tests. There's a test called the Fadibus test, which just uses a certain chemical and a certain reagent and it reacts with a certain thing in saliva. In saliva, you have something called alpha amylase. The alpha amylase in saliva, this thing will react with, and you can tell if it is saliva or not. And the Fadibus test is presumptive. We do it right there at the crime scene. When you then couple it with a rapid stain identification series, or RSID test, it confirms that it's human saliva. So what then? Well, then we send it off to a lab where it can actually be tested for other markers that help you to identify it right up to a subject. It's kind of like blood. It's nowhere near as precise as blood. We would rather have some other type of, you know, we would rather have some other type of marker, but sometimes saliva is all we can get. And finally, we can identify semen. And semen is actually produced very similar to blood. The cell structure itself is incredibly similar to blood in terms of testing, and so it can be identified right to the person. You can be identified based on semen left at a scene. And when the crime is a sexual offense, or maybe semen present at the crime scene, sometimes it's found on the victim's body. It could be in the clothing, towels, upholstery, and you can actually identify it presumptively just by seeing it. Crime scene detectives can see a fluid and go, yeah, that's probably semen. We need to collect some of that and send it off. Dark stains that may, when they dry, they may appear different. Those could also be semen tissues left over. So there are ways in which you have to collect it. You kind of dampen it a little bit, and then you swab it onto a cotton swab. And then of course there is the Christmas tree stain. The Christmas tree stain is the confirmative test that we can send it off to the lab, and it tests two reagents that are found in sperm. We can confirm that it's sperm. And then there's the Kernick-Trott. I hope I said that right. It can identify sperm based on making the head of the sperm react. It's a protein that reacts with another protein and it turns pink. And the picro-indigo-carmine, the picro-indigo-carmine reagent produces green on the tail, and couple that with the RSID and it confirmed that it's human semen. What can you do with an understanding that semen is present? You can tell that it was a sexual crime, okay? And if you then couple that with other things that put a perpetrator at the crime scene, you can support the claim of rape or sexual assault. Okay, that's it, I think. Wait, let me check just to make sure. Yep, that's it. So what I want you guys to do is make sure that you are completing all the assignments this week. There was a change in assignments last week, if you didn't notice, where I asked you guys to do a web quest. So you need to go back and do the web quest from last week. There's this and all the assignments this week, but I hope you guys found this interesting. Please watch the video that I've attached to this week's calendar. It's about blood splatter, and it's really well done, and I think you'll enjoy it. And make sure you get the mastery test done and complete all the assignments this week, and then I will see you guys next week. Happy Halloween.